Hey everybody, welcome back to Wednesday 8 p.m. Q&A with A&V. I am Vincent Racaniello, and sitting over there is Dr. Amy Rosenfeld in Columbia University. Hello, Amy.
Hello, I'm A.
How was your day? Was it fair, good, excellent?
Why do I hear it repeating? That's weird. Why do I hear it repeating now?
It shouldn't be repeating.
Well, it's repeating it in my headphones. It's weird. I don't.
Well, the we were just chatting and it wasn't repeating,
I know,
right?
but it's now. No, but now it's in my ears.
Okay, and is anyone hearing an echo? The audio here is correct on my end. You're still hearing an echo?
Yeah. Well, it's repeating the whole conversation.
Well, shut off the chat. You have a YouTube window open. Turn it off, okay?
Oh,
Just
okay.
close that. Yeah.
All right. That's better.
Yeah, right. Amy had the chat
Okay.
open on her computer. Okay, so good.
This is
I'm glad
better.
I was able to
Yes.
figure that out.
Yeah, me
So
too, because it was
here's an
weird.
interesting question. Oh, well, so Lost Laboratory said I just read Fever, and
Yeah, I read that. It's a great book.
I read that when I graduated from college, and I didn't know what I wanted to do. I was working in a lab, and I read it, and I thought, wow, viruses are cool, and it kind of pushed me to a career in virology. When did you read it, Amy?
Read it as a graduate student, way after I entered your lab.
So, Fever, you can get, reprints or whatever. You can get, yeah, I would highly recommend it. And yes, Westfield, I hope the pandemic inspires kids to go into virology. In part, that's why we do this, Amy and I, right? We want to inspire people, correct?
For sure.
For sure. Why did polio pique your interest? So
You
here's
had
how
no
it worked.
choice.
I had no choice. Amy wouldn't, if I said something different, Amy would call me out, right?
Yeah, you had no choice. You were
So
told
you do.
exactly what your postdoc project was in the
I was
parking
uh,
lot.
I was a postdoc with David Baltimore, and he said, "I want you to work on polio. I want you to clone the genome and sequence it and make an infectious clone." And that's what I did. And typically, what you do as a postdoc, you then continue in your lab. So that it didn't pique my interest in the beginning. It piqued David Baltimore's, but eventually I came to love it. And now even Amy works on poliovirus sometimes. Sometimes, right?
Yeah, for
Yeah.
sure. David had
So
been, but David used it as a graduate student. Part of his stuff with Mango was he did with polio. He was a RNA replication person.
Stanford scientists post entire sequence from Moderna vaccine. I don't know why that's unusual because the spike is out there. I don't know what this Stanford. I learned from you that the secret here is the lipid nanoparticle. Yes. I mean, I think they've published those formulations too, right, Amy?
Yeah. Oh, the lipids
Yeah,
composition of the lipid No, that's proprietary.
they have not.
Not
Yeah,
to my
so the
knowledge.
sequence
That's
is just the
proprietary.
spike. The spike was published way back in January last year, and anybody can have that.
Mhm.
But Amy told me something very interesting this week. You know, many of these vaccines encode a pre-fusion spike where they added 2 prolines, right? And now apparently some people are adding 6 prolines,
Yes.
and it makes the protein more stable, and you get more of it. So maybe it's even better.
Well, we'll see from the Newcastle trial in Thailand.
That's where they're trying it.
Yeah.
So they have at Mount Sinai made a Newcastle disease virus vector. Newcastle disease virus infects birds. It's a pathogen of chickens, actually. And they put in the spike of SARS-CoV-2 with 6 prolines. Remember early on, Amy, Peter Palese complained that nobody would pay attention to that vaccine that he had made?
Yes.
Maybe
I
adding more
think,
prolines is the key.
yeah, but I think for some reason, I think he's kind of footing the bill for the trial or something. I'm not exactly sure, but I don't think it's.
Amy, this is a question for you, Amy. What physically determines genome sense?
The Baltimore ski.
So if it's a plus, it's the same as mRNA. That's the rule, right?
Right.
And if it
If it's
is,
a negative, then the mRNA is the complement, the reverse
that's right.
complement.
So if you have a DNA genome of single strand, if the strand is plus, it's mRNA sense, and if it's minus, it's the opposite. And of course, if it's double stranded, you have both plus and minus. But
Yeah.
I don't know if physically determines is correct. It's just whether it is mRNA or not, right?
Right.
Here's an interesting one. Karate says the average person who recovers from COVID has antibodies that are just decent, but that's enough for this virus. Do you think that's why all the vaccines seem so great, that the bar is low? Do you know anything about this decent assessment by Karate?
I think he said it on Twiv. I think, it's average. It's what you expect. Like it's not like blowing you away. And they're just run of the mill antibodies. And that's sufficient to neutralize and block infection. I think the vaccines are great not because of the antibodies, but because of the T cell response, which we don't really appreciate.
Hello, Amy from an Albanian in Kosovo. You are an idol of mine. You're so smart.
That's cute.
Look, it brought a smile to Amy's face.
It is. Well,
You know,
somebody wrote here, Sharon Pfeiffer at Judy McInnes. Amy loves shoes. Like, I guess they must have listened to Susan Weiss and the shoe phone conversation.
here's a question from Raphael. The fact that variants emerged in different countries at the same time with some of the same changes could mean that the virus doesn't have that many viable changes in its repertoire.
Could be just hotspots too.
Yeah, hotspots and certain changes to make it more fit. We'll see as it goes on if that's it or if we get more changes, right? I asked Paul
Yeah.
Benash and he said he had no idea. We'll see.
Well, he's honest, which is good.
And here we have another person who thanks Amy. I'm highlighting this because I want Amy to realize that she is
Okay.
appreciated.
It's okay. Somebody
Do
is
we?
a new owner of a Twiv mug.
Oh, I didn't get to that one yet. Do we get any protection from adenovirus in an adenovirus vectored COVID vaccine? It depends. Not for the chimp adenovirus of AstraZeneca, because you usually don't get infected with chimp adenoviruses. But the J&J is a human, so you would get some, I suppose you would get some protection against that, right, Amy?
Well, yes, that's the whole problem with gene therapy, right? Is that the vector caused an immune response, and then when you came back with it again for the boost, the immune response went crazy. That's why the people died.
Yeah. Once inhaled, how long does it take until a SARS-CoV-2 virus particle successfully enters a cell? Is it minutes or hours? Do antibodies need to be faster than that to prevent infection? Yes, they are. I mean, it's minutes. Viruses get into cells in minutes. So unless you have a high titer of mucosal antibody, you're not going to stop infection. But later, the antibodies can limit it along with T cells, right, Amy?
Mm-hmm.
Is an activated virus better or mRNA vaccine better against variants? Any idea, Amy?
Well, an activated virus is going to have a lot more epitopes, right? And the variants that we know of are just in spike. So the mRNA vaccine will have to be reconfigured more frequently or, more rapidly than that inactivated virus. Because the inactivated, you're going to have thousands of other epitopes.
Yeah, I would get an inactivated vaccine. I thought that it would be good because it has other proteins besides spike, right? And as Amy said, those are T cell epitopes. So while COVID cases are going up, oh, I love the Mickey. Don't you love it?
I do.
But vaccination as well, or hospitalizations going up. Younger folks seem to be catching virus, and they do better than I thought. You know anything about hospitalization rates
They're
compared
going
to
up, but
infect?
it's all the younger people.
So last week in the U.S., deaths were down 21%. So hospitalized went up 4%. So not that bad. Yes, and because it's in young people, right? There's less hospitalization. Is that
No,
correct?
the hospitals are going up. You talk to Daniel, listen to Daniel. He talks about it. He says that it was a misconception. Theoretically, it was a misconception to say that younger people don't get severe disease. The majority of people in the hospital are younger people.
Does that mean now as opposed to previously? Have
Yes,
we gone through the old?
previously.
So we've gone
Well,
through the older people.
they died or they're vaccinated
Yeah.
or they stay home. Like my parents stay home. You guys stay home. But your children gallivant around.
Mm-hmm. Is that called
So,
misbehaving?
yes.
Ooh, here's one. What makes a virus lethal? Why are Nepa viruses so lethal? Could Nepa evolve to be transmissible airborne? That's three questions.
Well, there's multiple ways that a virus can be lethal. There's not just one mechanism to lethality.
So you could have high reproduction titers. You could have reproduction in many different tissues. You could have antagonism of the immune system. And Nepa, spills over from bats. It's not adapted to humans at all. And it kind of goes crazy. So it's reproducing a lot. It's reproducing in many tissues. Could it evolve to be a transmissible airborne? In theory, I suppose, but Ebola virus has been infecting people for many years and it hasn't done that. So I think there's some constraints. Same reason that
Mm,
avian H5N1 influenza viruses have not done the same thing.
I'm sure.
Are disease from other viruses as variable as SARS-CoV-2 with COVID-19? So I don't
Mm.
know, by variable, you mean different outcomes, different clinical outcomes? Um, so, the COVID has the two phases, the viral phase and then the inflammatory phase. And that's not common, but it happened with SARS, it happened with MERS, and it happened with avian influenza virus. So we have seen that before.
Well, there are other ways to be variable, right? You have the respiratory phase, and then you have the neurotropic phase. You have the gut phase of polio, and then you have the neurotropic phase, right?
Yeah. Yep.
So,
Is there any correlation between people who get vax side effects and those who were symptomatic? I know
no.
4 people who had COVID last year and their vaccine side effect mimicked their symptoms. No, I agree with Amy. There is no correlation. That's probably a coincidence. How do T cells destroy an infected cell? They release perforin, which is a protein that makes a pore in the infected cell. And then they release granzyme that goes through the pore and causes the cell to self-destruct, go into programmed cell death or apoptosis. Hi, guys. Now that vaccines are under EUA and placebo groups are gone, how can we monitor for uh, vaccine-associated severe, you know what I mean, adverse events?
Yeah.
Well, that's kind of the problem with the AstraZeneca vaccine, right? You're having these clotting issues, and there's no placebo. So the best you can do is to say, is what we see higher than we would normally see it in the population, right?
Right. And it's not.
What,
It's not any higher.
Amy? Do you say no to the vaccine passport?
Um, do I say no to the vaccine passport? Well, I'm a little confused about the vaccine passport.
What? Why are you confused?
So, so when you enroll your kid in kindergarten, you have to prove that your kid was vaccinated against measles, mumps, rubella, polio, DPT, blah 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 blah.
Yes.
So I'm not really clear why this would be any different. Um, that you should state that you were vaccinated. And
It depends how you use it, right? Like if you needed to get in a restaurant or if you needed to travel. When I was little, we needed to have a smallpox
why
vaccination
you have
card to go
to,
to Europe, right?
right. And you need a passport to go to Europe,
Yeah. But
right?
not to go to your local restaurant though, right?
Right, but you need to wear shoes and socks and pants and tops to go to your local restaurant.
Well, I think
And you need to drive in order to drive a car, you need a license. You can't just get into a car and not have a license. I mean, I guess realistically you could, but it's frowned upon, right?
yeah. I think to
So I'm
travel
not
a
really
passport is
clear.
good.
I'm just not really clear why this would be any different than any other thing that you do for any other vaccine. Or
Okay.
why it's different for when you get a license to drive a car or a permit. Like, I'm not really. So why is why is it different?
Um, I don't want to support or go against it, but people say they want a right to decide if they're going to be vaccinated or not. They decide whether they drive or not, right? If they want to drive, they have to get a license. But that's the difference. I don't agree with it, but I
Well,
think
the thing is that you don't you have to, in order to enroll your kids in school, and you have to have some kind of education. I believe you have to have some kind of education for your kids, and I think you have to fill out a ton of forms, although Pat Schloss probably knows better, like all the rigmarole you have to do in order to homeschool your kid. So if you are going to the public schools or even the private schools, you have to demonstrate that your kid is vaccinated. Why are you allowed to threaten my health? I just don't understand why this one why what you do for this one is so very different. That's
yeah,
what I
I
don't
agree.
understand.
Seatbelt laws. Yeah.
Right. I just don't understand why
Yeah.
it's different. I mean, it's
Okay.
like you can't go to the grocery store without socks and shoes. Well, not socks, but without shoes and pants on, right? And theoretically, you also like.
Well,
I can't remember the last time I saw in the summer a man in a grocery store without a shirt. So I don't understand why, if you do that, why wearing a mask is so horrible. See, like we have no problem with the others, so I don't get it.
Amy, are there any recent discoveries from your studies of polio? They'll be coming out, right?
Yes, they will be just coming out. They
It's
need
a
to
work
be
in
fleshed
progress.
out. It's a work
We're
in
working on it.
exactly,
Yeah, we're
exactly.
doing a bunch of experiments that are just beginning, and stay tuned. Do
Come
people
back in
who
a
do,
year.
maybe less, do people who do not have side effects from the vaccine possibly be asymptomatic and infected? Possibly, but there's no
No,
correlation, Elena. That it doesn't correlate. Daniel Griffin has said this many times, and one doesn't have anything
this
to
is
do with
just
the other.
this is just a rephrasing of her original question. She must not have liked our answer.
Yeah, hit like, folks. Just a little button down below. It helps us. Is there an immune escape variant already as claimed in India? I don't know what you mean by immune escape. If there are epitope amino acid changes, yes. But is complete escape? No. There's no immune escape variant anywhere of SARS-CoV-2. Do we have any more insight into if long COVID can happen to fully vaccinated people? You know anything about that, Amy?
I would assume long-term COVID cannot happen to fully vaccinated people, because the idea is that At least some of the preliminary data is that vaccine is protecting you against infection and transmission, right? But
Yeah.
that's done when your antibodies are really high, right? So we really don't know what it's going to be like in a year when your antibody levels drop and you have to rely on the memory response kicking out.
Yeah.
So I don't know.
As Amy
Ask
would say,
us in
ask
a
us in a
year.
year. Ask us in a year.
Yeah, come back later.
Cute dog. I have a relative early 60s cardiac issue being offered AZ, but she doesn't want to take it. Is that reasonable given the recent UK EU announcements? What
Just
do you
take it.
think?
I would take
Just
it.
You would take it?
Mhm.
And another question from Judy in Canada. Would you take the AstraZeneca? I would, and Amy would also. Yes, I'm 68. Well, it's for younger people. They don't want to take it, right, Amy?
Right, but I'm younger than you. I would take it.
Why is the media saying variants are more deadly? Simply because it's more contagious and can make more people sick, and not that the virus makes them sicker? No.
Well,
There are a
I
few
don't think it's
studies.
making more people sick. I think it probably replicates more efficiently, and so it overtakes the population. So it's harder to find other forms of the genome, and so it looks like it's making more people sick or whatever. But I don't really quite get it. But that's what I think.
I put two links to papers at the very beginning of the chat. One of them is a study looking at the B1117 variant in hamsters, showing it's not more lethal than the other isolate. Thank you, Michael, for your super chat. Much appreciated. Goes to the new studio. Hopefully opening next month. Amy loves shoes. Do you love shoes?
I do. Shoes are great. Shoes are fantastic. There's nothing like a really pretty pair of shoes.
Scientific briefing today about AZ suggests younger people mount a different immune response. Could this rare event tell us something new about the virus and ACE receptors?
I didn't see it.
Yeah, I don't know. I haven't seen it. Sorry. We'll take a look. Come back next week. Very Nika, thank you for your super sticker. Appreciate it. And here's the proud new owner of a Twiv mug. Wonderful. I don't even have one here. Do you have one?
Yeah, but I'm in a lab. You're not allowed to have it in the lab. But yes, I have one.
Have you heard the phase 2 trials of a nasal spray in the UK? 99% viral load reduction, not a vaccine, but nitrous oxide. Do you get like lightheaded as a consequence? That's what the dentists give you. I don't know about it.
I don't know, but this goes along with the lines of some work that Michael Diamond published.
How many days post-vaccination will you have appreciable levels of antibodies produced to be at least somewhat protective? Amy?
Need a minimum of 2 weeks. But that's define appreciable. Detectable?
Yeah, it's somewhat protective. You want to be protected against dying, right?
Right. But I would say you have detectable levels of antibodies in 2 weeks.
Yeah.
No sooner.
So in the some of the trials, they do a curve of
it's an average
versus vaccine, and they start to count the cases with time, and the two curves separate. It's a count of the cases, Amy. The cases, positive cases in each group, and they start to separate at day 11. Amy fan from Houston, thank you. Since all COVID vaccines present the immune system with almost the same spike protein, shouldn't any be okay for a second dose? Why should they have different efficacy?
They should all be okay for the second dose. They have different efficacies, probably due to the uptake of the particle. It's not always equal.
Yeah, like the nanoparticle versus a virus vector particle.
Right.
So they could be different, yeah.
But even the nanoparticles the lipid membrane is composed differently.
Right.
So,
could a coronavirus mutation make the coronavirus stop causing COVID-19? Sure, could. Maybe 10, 20, 30 years. Maybe, right?
Sure.
Like become a common cold coronavirus. We'll see. I don't know if I'm going to be around in,
Well,
how
I
old
don't
am I?
know that the
68,
comment. Yeah. 30 years, you'll be
20.
98.
Will I be
But
around?
yes. But the thing is I don't agree with what you just said. That the
What?
mutation is going to make it a common cold coronavirus. No.
Well, I don't think we
Us
know.
having immunity, and very young children being the
I
sole
agree. Agreed.
source of infection is going to make it a common cold because we've observed that that population less frequently gets severe disease.
Okay. I agree. Corrected. How consistent are the peptides that result from the proteasomes chopping up spike from mRNA or vector?
They're
How
all different.
consistent? Yeah, they're different. And from person to person, they can be different, right?
Mhm.
Because the MHCs are different, so the peptides that are displayed are going to be different. That's why T cell immunity is resilient.
Seti and Cruddy listened to TWIV 2 weeks ago.
So Overjoyed said she had multiple infections and no antibodies. Maybe the vaccine will do it. But
Maybe.
are deaths going down in the U.S.? They're not, right, Amy?
No, they are. You just said they were. Hospitalizations, not.
That's right. That's correct. And I got the information from the New York Times tracker. So in the past week, cases have gone up in the U.S. 19%, deaths down 21%, hospitalization up 4%. Those are the numbers.
Yeah,
Amy is appreciated. Love the straight talk.
it's good.
Oh, do you see children's vaccine as an absolute prerequisite for ending the pandemic?
Yes.
Yeah, I think the goal in the U.S., what is it, 15 and up by the end of June? Is that what Daniel said? Or is it 12 and up?
12.
12
12 and up. And then by the end of December, closer to like 6 months.
Okay. So we have, no, I do not listen to the weekly Osterholm podcast. Here's why.
Because some of the stuff he says is totally wrong. And I don't like to stress myself out any more than I already do.
Yeah, and I want to put time into my content and not consume others. So I don't. I feel that I am able to create my own content without the input of Osterholm and other such podcasts. Why are people so scared about these vaccines? I usually like showing people your answers. That's why I ask. What do you think, Amy? Why are people scared about the vaccine?
Because it's been marketed poorly.
By who?
Everybody. Scientists, everybody. It's been marketed poorly.
So how would you
See,
change it?
you can't start off a vaccine with we're going to use a novel technology that we don't know anything about. That's not a good marketing strategy. That
Did you
that
hear?
embraces fear.
Did you hear the TWIV discussion where the package insert is like, this is an experimental vaccine. You might still get sick, you know. Of course, that's a package insert. I would say
Yeah.
Santiago, that for most of the vaccines, many millions of doses have been given. They're protecting people against severe disease, and they are safe.
Right,
That's
but
the
that's
best.
not why people are scared.
And
People are
Amy and
scared.
Vincent both got vaccinated, and that's why you shouldn't be scared, right?
That's true. But you need better marketing.
Surely spike mutations 50 and 501Y, E44, etc., represent an evolutionary maximum allowing better cell entry. I don't know if it's better cell entry or just evasion of antibodies or some other aspect of enhanced fitness. But yes, they are being selected. It's clear. And we don't know yet, but hopefully in a few years, people will do the right experiments. Okay, Amy, what's the
Really?
average
You
day
want
like
them
for?
to do the right experiments? Why
Yeah,
start
well,
now? Why start now?
Amy, what's the average day like for a virologist?
Busy. Busy. Ah, let's
You,
see.
you get up and you come to the lab, right?
Well, you always have to have coffee. It's
Yeah, well,
barbaric without any coffee. That's just
then you,
barbaric.
what's the first thing you do? You start doing experiments. You think of what you have to do for the
Um,
day.
I think of what I have to do for the day. I look at plates. I do some reading. Um, then I look at a lot of cells. So I go to the incubator. I see if certain cells need to be split. If I have how my plaque plates are looking, and various other things. If I have infections going for stocks, I look to see if I have 100% CPE and whether or not it's time to freeze thaw and start the ultra. Um, and then a few meetings, a lot of reading, a lot of experiments. Then I visit the mouse house. Then I come and talk to you sometimes. Then I visit another mouse house. Then I go to a lot of mouse houses. What can I say? Then I do plaque assays, make new plates, do some PCR, some RNA extraction. And by that time, I don't know, between 8 and 11, and it's time to go home.
I won't tell you what my day's like because I'm very different. I do this communication stuff. I don't do experiments. The only thing I do is I help Amy with the mice. I change the CO2 tanks and the liquid nitrogen tanks for Amy, right?
Yes, since I broke my foot, I'm no longer allowed to do those
She
activities.
cannot change the tanks because the last time she did, she dropped it on her foot and broke it. And I told her, if you ever do that again, I don't remember what I said. I said, don't do it
Yeah,
again.
yeah, pretty
So
much.
sometimes I have to come in on a weekend to do it, right?
Yes.
If viruses get into cells in minutes, how can a vaccine be sterilizing? So most are not. There are very few, in human papillomavirus vaccine is because it maintains very high levels of mucosal antibodies, apparently, to block the virus from getting into cells. But maybe still a few get in, but it's not enough to detect. Okay, most of the others are not. Any thoughts on the Novavax? The results looked good. We're waiting for them to present their phase 3 to the FDA. That should be happening any day now. I thought it would have happened already, actually. Hello again from Wisconsin. General public now eligible to get vaccinated. Very good. Osterholm still seems convinced about another bad spike. So Osterholm said it would be the worst month ever in March, and that didn't happen. So I'm not sure he's a good predictor. Could an mRNA vaccine be made to code for more than spike? You bet. And it may be that second generations do code for other proteins now that we realize that T cells are crucial. Right, Amy?
Right.
It was Minnie. I'm sorry. Okay. I don't know my characters. Did Daniel listen to Arturo Casadeval? Probably. He listens every week.
He
Listens
retweeted
every.
it.
He did?
Yeah, he retweets it. Yeah. But I'm not sure I would have said terrific.
Is it
Lots of
really okay to
issues.
switch vaccines for the second shot? Well, you can't because the FDA hasn't approved it here in the U.S. So
But
technically
Mexico, they
not.
can do whatever they want.
Yes, in Mexico you can do whatever you want because you can get drugs at the store without a prescription. So I think it's fine. I think they're compatible. The reason the FDA doesn't want to approve it is they haven't been tested, mix and match. So, but I do think the logic that Amy used earlier is enough.
Mm-hmm.
I agree with Amy. Nature published an interesting preprint where new imaging diagnostic techniques think that once you progress to severe age, no longer matters.
I don't know what that's in reference to. Do you?
No. In lecture 22, you mentioned SARS-CoV-2 may have not originated in China. What other origins? Well, as Amy will tell you, Lympho1 found very similar viruses in Thailand bats. And then some have been found up in Japan, right? So 4,800
It's a
kilometer stretch.
it's a 4,300 square kilometer distance. So I think, and people put things on cars and they drive them through the country, and they also drive them internationally. Like I used to drive between here and Montreal. So let's say I got sick when I was visiting my parents when I was a postdoc in Montreal. Does that mean that the virus came from New Jersey? Probably
No,
not, because I was
no.
living in Montreal at the time, right? But you would have
Yeah.
focused in on the fact that it must be circulating around Essex County, New Jersey, because that's where I was diagnosed. I think people should think more broadly, or I don't know. I just find that it's very myopic.
Well, Jan wants to know how the vaccines work against P1. I know that some vaccines tested their vaccines in Brazil, but I don't know. And I think some of them did well, but I don't know which one. It may have been the J&J. So some do, and I'm not sure all of them have been tested. Do you know, Amy?
I believe that Pfizer, Moderna, and J&J have been tested against the P1 variant, and I believe that you do see equal protection because you're in people, because you're accounting for T cell epitopes and other mechanisms of blocking or resolving infection. Whereas in your tissue culture plate, when you're just doing neutralization, it's just one aspect to protection.
A coworker received J&J vaccine, wants to receive Pfizer instead. What would happen? It's fine.
It'd be fine, but why are they getting Pfizer when J&J is one shot?
Because they already received J&J and they want to get another one. I don't know.
Go ahead.
It's fine. What is molecular mimicry? So let's say a virus has a protein and a little bit of it looks like a human protein. So when you make an antibody against the virus protein, that antibody may react with your protein. That's molecular mimicry.
So somebody must be, so this Francois Dufour must be really from Montreal, because they want to know what the favorite bagel shop is in Montreal. It's a really funny question, because it's like a rivalry between St. Viateur and the Fairmont. And nobody takes sides. That's just ridiculous. So they either live in Montreal or something or other.
Okay. What are the chances of insertional mutation using adenoviral DNA vaccines such as AstraZeneca or J&J? Would these insertions be random, and what are the chances of activating proto-oncogenes with such insertions?
Well, the viral genome is gutted, number one. And number two, adenoviruses do not integrate. So I don't know I don't really understand. If you understand the life cycle of adenovirus and you understand how it works, I don't understand where you would come up. I just don't understand this question.
And in addition, the adenovirus vectors do not replicate, so there's very little DNA getting into the nucleus, which even reduces even more the chances of insertion. So I think it's very low.
Yeah. But it doesn't insert. It doesn't integrate.
Doesn't integrate. It's reported Vancouver NHL hockey team and family members have tested positive with P1. I'm not surprised. Viruses travel around the world. And so that's what this one's doing. I don't think that's an issue.
No, you should talk to Daniel, because Daniel has said multiple times that he was consulted for the NHL COVID policy.
And?
I don't know.
Okay.
Yeah, okay. I'm sure he'll tell you. I'm sure he'll have more information if you ask him.
Jessica wants to know what makes SARS-CoV-2 so dangerous and highly transmissible compared to polio, smallpox, or Ebola. Seems like corona is a notch above the rest. Well,
I don't think that
SARS-CoV-2,
that's true.
it's not true.
I
Yeah.
don't. I was going to say, I believe that smallpox and Ebola are pretty transmissible, and Ebola mortality is like what, 50%?
Yeah, 50 and up,
Case
depending on
mortality.
the country. Smallpox is like 30%. What's the mortality in polio about? Do you know offhand? I mean, the fraction of the bulbar polio people die, like, but that's a small fraction of the total, so
Yeah, I
that's
think
quite low.
that's like 0.1%.
Now,
Um,
SARS-CoV-2
and
transmission is. Go ahead.
I don't know that I would say it's more transmissible compare. So what are we going to use as the transmissible index? The R naught?
I think that's all you have. Yeah.
So I would say it's on par with polio, and polio actually might be more
Yeah.
because you have 99.9% of symptomatics. It survives in the water. And it's, yeah, I would say actually the R naught of polio is probably higher, right?
Yeah. So I'm not sure, Jessica, it's so dangerous, really. 80% of infections are either asymptomatic or mild. Now, that doesn't take into account long COVID, I know, but just for that first phase. Oh, here's one for Amy. Does Amy believe that SARS-CoV-2 infects the GI system, or are GI symptoms purely immune response?
I have not seen a lot of convincing data that that it's in the stool.
Yeah.
That infectious virus is in the stool. It's clearly coronaviruses are enteric viruses in the bat.
Are previously COVID positive people more likely to react to a first vaccine dose?
What did Niche say today?
I think the first dose after an infection is like the second dose in a vaccination scheme. So, yes. Does the T-detect test differentiate between natural infection and vaccination? No, it just looks for T cells against certain that recognize certain peptides by virtue of their T cell receptor. So, no, it doesn't.
Excuse me. All right, it's 8:40. It's 8:43. 3 more questions, and then I have to go.
All right, next, here's one. How many SARS-CoV-2 virions are needed to cause an infection? My husband was appalled that it takes only 18 noroviruses to cause disease. That's from my lecture. I don't think we know, frankly. The reason we know for norovirus is because we can challenge people and give them different amounts of virus and see when they get sick. We can't do that for SARS-CoV-2. Although maybe they're going to do that in the UK challenge trial, right?
Maybe.
Are children getting 2 shots or 1? Well, if it's
Oh, they're
Pfizer,
getting 2.
2. Yeah.
I think
Go
that
ahead.
the was it Pfizer that said, or was it J&J that said like Moderna had like already put in the paperwork for 12 to 15, so they were going to do 6 months to whatever. Wasn't there something about that?
Mm-hmm.
So
Here's one for you. In previous work, various small molecules were studied designed to prevent the action of 2-O-methyltransferase. Today, do we have any candidate molecule for COVID? It's right up Amy's alley.
why is this up my alley? Not that I am aware of.
The methyltransferase is for the cap, right?
Yes.
And I know Amy, when I think of Amy, I think of translation always.
Yes, I spent a lot of time studying translation.
One
But
more?
not that one more. Not that I'm aware of. Plus, you would have to be very careful because you don't want to inhibit the host methyltransferase enzymes. That would be really bad. That's why you don't target things for translation.
How
generally.
concerned should a healthy 20-something vaccinated individual be about long COVID?
How concerned? Like never go out of your house concerned, or I'm aware it's a potential possibility. So I don't know what these gradients are. Like, is it like I'm never leaving my house?
No, I don't think you're that worried.
I don't think I'm not worried, but I think
we don't know. I think we don't know at this point. Okay. And I would err on the side of you're not going to get long COVID if you're vaccinated, but we'll see. It's just too soon in the game to tell. What do you think?
Yeah, I think it's too soon because the studies that show that it blocks transmission and infection, your antibodies are really high. It's not done when it's a really normal situation. So if you're blocked, and if the idea is that you get less severe disease, I do agree that a lot of the long-term COVID had mild symptoms, but what is less severe disease then? I'm not, if you say that at 20 I get mild symptoms, what is less severe disease that you're protected against? Do you know what I'm saying?
Yeah.
So I don't really know.
Take this last one, Amy, because it's for the A.
What drives this for the A? What drives the cyclic nature of a virus like EV68? So this happens to be a topic of discussion. But so while there was a Science Translational Medicine paper doing mathematical modeling about how they could predict retrospectively EV68 by biannual cycles, the study is significantly flawed for just based off of the fact that it was a biofire that readout was entero/rhino. The real idea is that you have a naive population that is immune naive to the virus, and then it gets infected, it drops the target population, and then you need to rebuild that naive population to a level of where you can spread the disease. And actually, this is most elegantly described in Peter Daszak and Ian's Nepa paper in the bats that was kind of glossed over when people who were discussing it were so mesmerized by the tracking devices that were inserted into the bats that they missed the whole point of the paper. It's true. They had a whole discussion of the tracking device, and I had to go, excuse me, the point of the paper is that infection works just like it does in humans, right? They had a naive population of bats that then were infected with Nepa. It spread throughout that colony. And then then you saw a depression of transmission and of Nepa virus, right? Until those bats had new bats and were targets for the infection, right?
Yeah.
And
so it's cyclical.
It goes up.
It's antibody levels, yeah.
Right. It's antibodies and T cells.
And you have new babies born, and get enough of those every few years, and then you have another outbreak of 68, right?
Right. And it's just like there's, what was it? What is it? 5 years for Nepa? Every 5 years you see an outbreak within the bat population, and then it overspills every 20 years into the humans.
Yeah. Yep.
Isn't that what the paper talked about? And then we thought it really had to do with seasonality or pregnancy of the moms, but it really had to do with the fact that there was this, now bat colony of let's say you had 100 bats, they all were protected, then they had 500 babies. You got infection swarms, right? And those
Yep.
500 babies didn't breed for a couple of years because they're not sexually mature. Then they have 15,000 babies and voila, right?
All right. Thank you,
All
Amy.
right. Thank you. I'll see you tomorrow.
Bye-bye. See you in the morning.
Bye.
Be
Yeah.
safe.
All right. Bye.
Bye. Okay, you're stuck with me for the next 40 minutes or so. If the virus came from the animal trade in China, most likely, wouldn't there be a trail of sick people? Text messages. Hey, I'm sick. I don't know. It's a good question. Depends how many. You know, the early stages of a spillover, it's not easy to pinpoint. And, you know, HIV spilled over in 1920 from chimps to humans, and we didn't detect it until 1981, right? So there you go. So I'm not sure. You might expect that or not. If it started in the country, here's the scenario, I think. A farmer is going into a bat cave collecting guano, fertilize the fields, gets infected, goes home, infects his family. The family goes into town. They infect a few more people. Nobody gets terribly sick, but the virus is moving around the population, adapting. And then one of those goes to Wuhan, maybe to the market, and infects a whole bunch of other people. And boom, we see the 41st cases of pneumonia. That's what I think. So you have a very quiet, silent spreading for a while, and then that's it. But we'll see. As I said earlier, we're going to have the three people who chaired the WHO Commission on TWIV. Peter Daszak, Marion Koopmans, and one other lady from Denmark whose name I forget, but she has been on TWIV also. Colston. Okay. Let's see what we have here. Any estimate on how long the vaccine might last? So if the viruses don't change to need reformulation, like a flu reformulation, I think years is fine, but I'm not sure. I think we're going to have antigenic variation that requires reformulation every couple of years maybe. So otherwise, I think they would be durable. There's a lot of discussion of passports here, but I don't want to get into that. Are variants being obsessed over due to the nature of the extensive surveillance? And do pseudovirus neutralization experiments really correlate to real-world vax efficiency? So let me take the last one first. So a number of people have told me that the pseudovirus titers and the neutralization with authentic virus, SARS-CoV-2 that is, are pretty comparable. However, it's only antibody, right? And what happens in a person, vaccine efficacy is a combination of antibodies and T cells. And we know that T cell epitopes are not changing. So that's why I don't worry about the variants. I think the surveillance, the genomic surveillance we have had during this pandemic is more than we ever had before. And I don't think we know what to do with those data. We see a variant emerge and suddenly it's in a lot of people. And so people think, oh, it's got to be more transmissible. But that's an association, right? That's what we say not to do. So I think we have to learn what to do with these data. Now, you know, a change in spike that affects antibody reactivity, we need to track that. But as long as T cells aren't affected, I don't think we have to worry about it. That's my take. And if you read The Street this past week, the article, I said it pretty bluntly. But most of the mainstream media doesn't talk to me. So most of the stuff you hear is the opposite. And then people say, why are you the opposite of the others? Because I've been thinking about viruses for 40 years. This is my idea. And I'm probably going to end up being at least partially right. We'll see. Let's see. More vaccine passports. Let's get through them. Long COVID. You have to ask Amy about that theory there. Yes, media is repeating what epidemiologists are saying about the variants. So the epidemiologists look at patterns, but they don't look at the virus, right? And that's what's missing. I'm a virologist. I look at the virus. And you guys, there are many of you out there say, you're wrong, Dr. R. but I'm not. We're missing data. And this data on more virulent, it's not holding up in animal models, at least for the UK variant. Is it possible by a specific test to know what vaccine type you were given? No, not most of the vaccines provide spike protein, right? And not other proteins. So if you look for antibodies to spike, it could be the vaccine or it could be natural infection. And then the inactivated vaccines have other proteins and nucleoproteins. So you could pick up antibodies to that, but you'd also have antibodies to spike. So there's no easy way to do it. Is there a way to trap virus inside infected cells so it cannot bud? Yeah, you could. You could inhibit the transport in a number of ways, but these are cell pathways that are essential to the cell. So you would end up injuring the cell, and that's not a good approach to take. You can't hurt the cell. You can't have side effects. Are you hearing B117 is 40% of new cases? No, yeah, I am, and I'm not surprised. As a fitness, it has a fitness advantage. So the virus arrived there in the midst of an epidemic, and it gets incorporated into that epidemic, and it just follows the previous isolate. It doesn't mean anything about that particular variant other than it competes well with the other. It doesn't mean that it's more contagious, which is what a lot of people are saying. I heard that 1117 is more stickier. So some of the changes may increase affinity of the virus to receptors. You can measure that. I don't know what effect that has on the infection. Nobody's really looked at that in a careful way. People make assumptions. They say, oh, here's a change in the receptor binding site of the spike. That must affect affinity. But they don't actually test it. That's what I'm railing about. That's what I rail about. Okay. Do you have insight as to why the FDA allowed the 1963 polio vaccine lots known to have monkey virus SV40 in it? So it was an accident. They didn't know that SV40 was contaminating these cultures until the vaccines were already out there and given. And a lot of epidemiological studies have been done which do not indicate that vaccine caused any cancer in people. So I agree with that. I've looked at that quite closely. So I don't think there is. Thank you, Mary, for your super chat. Appreciated. So we are moving into a studio in May, hopefully. Daniel Griffin and I are sharing an office/studio space where I'll be recording most everything except these live streams. I'll keep doing these at home because I don't want to stay in New York till 9:30. But it will enable me to focus on content. I will go there most days. I won't drive anymore. I'll take the train in. I'm really looking forward to it. And anyone in New York, come visit and get a picture, Yes, Amy is unself-conscious. She is very smart and doesn't sugarcoat. And she tells me I'm wrong. And I often say, could you be nicer? And she said, no, because if you're wrong, you're wrong. And tomorrow she'll give me hell about saying this. Some days, Thursdays, I come in and she goes, why did you throw me under the bus? I said, I didn't throw you under the bus. Oh, this is very interesting. Shouldn't older people get larger doses and boosters? As you know, for some vaccines, they do. The high-dose flu vaccine, chickenpox vaccine before the Shingrix, the same thing. Whether we need to do that for COVID vaccines is being looked into. It's a good point. What enabled coronaviruses to transmit from animals to humans? Well, random mutations occurring in the animal that just made a virus that happened to be right to grow in people, and then they encountered a person. That's the key. You need mutational diversity and you need an encounter, because you can have bats in the cave spawning all kinds of human infecting viruses, but if there are no humans around, nothing's happening, right? So that's a key part of spillover from animals, you got to have an encounter. And that's pretty random. But if you do things that encourage contact with animals, like you build a farm next to a bat cave, that can encourage it, right? In the Nepa outbreak in Malaysia, they put pig farms in orchards, fruit orchards. At the same time, they were cutting down the forest to make plantations, and that chased the bats out. They went to the orchards where the pigs were, and they infected them with their viruses. Well, you can find these scenarios. Listen to my lecture on emerging viruses. It's got a bunch of stories like that. I'm sorry, you're the only one saying my sound is bad. So maybe it's something on your end. I'm sorry. Can I take inactivated as a second after AZ first? I don't see a reason not to. As I said, in the U.S., you couldn't. You should probably wait a month to make sure you get the full response from the first vaccine. If you had a 13-year-old kid, would you send them to school without being vaccinated? Oh, it depends on the extent of infection in your area. So if you're in a rural area and there are no cases, sure. If you're in a city where there are a lot of infections, I'd keep them home. And that's tough because kids need to be in school. Yes, they are vaccinated gorillas. In fact, the person in the San Diego Zoo who is doing that is coming on TWIV in a week or two, Lisa. So stay tuned. No, thank you. We're not amazing. It's just what we do, and we like to share it with you folks. People are scared of the vaccines because they're brand new. That's part of it, I think. They are brand new. Give it a few years. I think we'll be fine not getting everyone, and then in a few years we'll be good. My friend got a pedicure, and her technician said her and her 4 daughters are not getting the vaccine because she heard it makes women infertile. Yeah, so that's a rumor that's going around that says there's molecular mimicry between the spike and a protein essential for the formation of the placenta. It's not true at all. It's false. It's a rumor, and it spreads. You know how these things go. So you should tell her it's not true. She can get vaccinated. I haven't listened to the Osterholm podcast. I'm sorry. As I said, I make my own content. I typically don't listen to other content, especially in my own area. You know why? I want to make my own conclusions. I don't want to be contaminated by what I hear from others. I don't know if you understand that, but I want to think on my own and make my own conclusions and put them out there. Any new science on the Ebola outbreak? No. I haven't heard anything. This is, thought to be a virus that had stayed in a person for 5 years since the previous outbreak. So I assume we're going to have a paper soon. Amy didn't test herself for antibodies, no. And neither did I, although you can buy kits now and you can do that. I can answer for Amy because she starts texting me in the morning when she gets to work. She gets in like by 8-ish. And she's there now. She's going to be there till 10 or 11. Thank you, Andrea, for your super sticker. Very, very generous. And as I said, we put it towards our studio. And I hope to someday hire people to help me do the technical work. I'm just tired of editing. I'd rather read papers and think, right? Oh, this is great. UK is doing rapid testing for everyone at home twice a week. This is great. This should have been done last year, right? That would have made an impact. Wow. How do you do PCR testing on mice? Like any other. You take a sample, whether it's blood or, nasal swab, rectal swab, pellets, fecal pellets. When you pick up a mouse, it poops right away. So you can just pick up the pellet and do a PCR, whatever. It's easy. And then you have to extract the nucleic acid. Of course, do I know governments who are researching viruses to be used as weapons? I don't know of any more. You should go listen to the TWIV with Jens Kuhn, because he went to the former Soviet Union after they disbanded their weapons programs, and he can tell you about it. But he's not aware of any going on. But really, if there were, would we be aware of it? I think not. What are my favorite pieces of lab equipment? So my favorite piece is the ultra centrifuge. It's a big machine, stands on the floor, it spins really, really fast. You put rotors in that spin, you put tubes in this metal rotor, and they can go up to 100,000 RPM. They're made of titanium, so they don't break. And they're really expensive, but you can purify viruses. That's just, I love them. Ultra centrifuge. We have one. We have one. What's the likelihood of someone being infected 6 months ago and then infected now with a variant? Okay, so you'll get infected. Probably you won't get sick. You certainly won't go to the hospital. You won't die. Okay, that's what's going to happen with a variant, as far as I know now, unless they keep changing and they really overcome immunity. But right now, the T cells save you, and the antibodies are enough to keep the titers down. So I'm not concerned about that. What is the most lethal rabies virus? 99.9% lethal if untreated. No question. Do vaccines with protein-induced pre-fusion spike prepare antibodies any differently? No, they don't. The pre-fusion just gives you more protein, so you get a better immune response. The AstraZeneca vaccine had very low efficacy in South Africa. That's correct, and they decided not to distribute it in South Africa. But elsewhere, it has been working. You need to make a drinking game when Vincent says, right, Amy, all the guys drink. When Amy says, I don't understand, the women drink and everyone drinks on variant and epitope. And then you're going to all be drunk in no time, right? That's very good. What are your thoughts on long COVID? Is that something an individual should be concerned about? I don't think you can worry about something you have no control over. I think you should worry about not being infected, which you can do by wearing masks, distancing, and all that stuff, right? I have been uninfected for a year because I behaved properly. Now, I'm not a frontline worker, so frontline workers I understand. But people who can choose to stay distant, it can be done. And yes, I do. Osterholm is sometimes right. I have respect for his knowledge, for sure, but he's not always right, and he's overdramatic. I guess I can be overdramatic too, right? Can babies be born with COVID? In other words, can the virus cross the placenta? People are looking into that. There is a study ongoing. We don't know yet. At least I don't know. Will Nova vaccine be effective on the new variants? I think it will prevent severe disease and death and hospitalization, yes. But you can change it in a year or two if you need to. Very Nika, thank you very much. Now, you had a question for Amy, who's the PCR goddess, right? But she's not here. Let's say a cat was tested for SARS-CoV-2 and the PCR came back positive, CT40. What could that mean? 40 is not, it is very little RNA, not infectious. It's either a false positive or the very, very end of an infection. That's what that means. Lin Fa Wong, well, don't think so. He barely answers my email. He's a busy guy, but he did do a couple of twivs with me. You know, maybe once this is over, people like him will have more time. But hey, we've got some good guests, and we're getting, as I said, 3 people from the WHO committee who are cool in their own right. So, oh, favorite bagel shop, there it is. Are the pathological consequences of viruses due to cytolysis, releasing more viruses and damaging tissues? Well, viruses can get out of cells in many ways, not just by lysing cells. They can bud out of cells. So that's not just the only determinant. It's one, yes, if you kill cells, that's going to contribute to pathology. But immune responses are as much a contributor to pathological effects of viruses as does the virus themselves. I make a big emphasis on that point in my lectures. Immunopathology is just as important, if not more important. For some viruses, they don't kill cells. And all the pathology is immune driven. Yes, so the reason the press believes the variants are worse, Osterholm goes on every network and says, yes, it's more contagious and more severe. And these numbers, by the way, come from early studies, which I think were flawed. And people just keep repeating them without looking at the original data. And if you go and look at the data, I guarantee you, you will be suspicious. So, yes, that's what's going on. And they don't ask me. I don't go on every news channel. Yeah, it's fine. I'm telling you guys, you heard it first. UK decision to delay the second shot is fine. We looked at the study that informed that a while ago. The data are good. You still get rising antibodies after the first for some time, and so you can delay the second shot. I think that's fine. A lot of people think it's not good, but their data said antibody levels continue to rise, so I think it's fine. Someone has HIV with a CD4 T cell count of 100. Now that's AIDS. Anything less than 200 cells per ml is AIDS. Should you get a COVID shot? I don't see why not. But again, you're not going to make much of an immune response, right? I don't think there's a counterindication. And you could get the shot, and if you got sick, you'd have to get monoclonals, right? So infection fatality rate really depends, important, John, on where you are. It varies greatly according to healthcare system, country, and so forth. So, if you just take the numbers, so let's, we could do that. We could take the numbers, and you guys can do the math. I don't want to pull up a calculator. We have, 2.8 million deaths globally. We have 132 million cases. Just do the math, and you can come up with it. But that's a bogus calculation because it's an average of the whole world, right? Talk about the double mutant variant. So this is nothing. There are double, triple mutants everywhere. That just, these spike changes are accumulating. And they're going to be popping up all over the place. And, when these variants are more fit, they have a reproductive advantage in the population. And why that is, we don't know. So, yeah, if they get to California, they're going to spread. But, the previous virus would have spread as well. So this is just displacing it. I don't see any difference with the variant, but people don't see that for some reason. If you already had COVID, could you still pass it on to another if you have recovered? No, if you're so many days from symptom onset, 14 days max, then you're not going to be infectious any longer. So that's why we don't have to be quarantining after that time. Alan Townsend, that's a good idea, sure. I tested positive for antibodies in March 2020. Was I infected, or could it have been cross-reactive antibodies from another corona? No, I don't think it's cross-reactive from another corona. It depends on the test. What is Abbott checking for, spike or N or both? So if it's both and you were positive for both N and spike antibodies, then you were infected. If it were testing for both and you came up with only spike, then you were vaccinated. All right, I saw this question earlier. How does an envelope virus survive the gastric pH? So yeah, that's a good question, because we were taught when I was learning that envelope viruses don't do well in the GI tract, but coronas are different, right? The porcine epidemic diarrhea virus, severe acute diarrhea syndrome of pigs. Yeah, they're enteric pathogens, yet they're enveloped, and there are others too. So somehow the structure allows the virus to survive. How that is, we don't know, but it certainly is different from influenza virus, which will not, reproduce in the GI tract. Most strains won't. Some of them will, but most will not. Is it safe for my daughter who breastfeeds to get a vaccine when she and her baby had it in early December? Yes. And if she will pass the antibodies to the baby, that's fine. That's fine. People are saying that's okay. I've heard Daniel Griffin many times. And yes, vaccination seems to improve long COVID. The question is, if you don't have long COVID and you get vaccinated, then you get a mild case. Will you get long COVID? And we simply don't know that. Why don't we see many spillover events from domestic pets to humans? We don't look. It may be that you test positive and you got it from your pet, but nobody's going to check your pet. And you might not know the difference. So that's what I think. We're not looking at pets very much. We should. If the virus mutated the same proteins in different countries, that means, oh, we answered this already. Sorry. Vincent, the big question tomorrow in your AMA is going to be about why B117, blah, blah, blah. Fine. I have no problem answering that, but I think that's boring. There are more interesting issues. If people really want to focus on that, it's their AMA. No problem. I don't, it's hard to compare countries. So Chile, I don't know the actual extent of vaccination and seroconversion there, but in Israel I see the numbers because they publish them. So I'm just not sure. Isn't the real variant of concern the ancestor of SARS-2 variants dated in the recent WHO report? There's no common ancestor. Well, yes, there's a common ancestor of SARS-CoV-2 and then of RATG13. That's correct, which circulated some time ago. But between that and SARS-CoV-2, the real ancestor lies. And that, well, you know what? There are other viruses out there that are going to spill over. So they're all concerned, which is why we have to figure out how this one got here and try and prevent the next one, right? So, SARS-1 was in an outdoor meat market. We knew that. And civets to humans in meat markets, so they closed the meat markets for a period, but then they reopened them. So, you learn how it's transmitted and then you go back to it. So that seems to be a problem. There's no evidence that that's the case here. Emphasis. But we have to find out where it arose and do something about it. I don't know who Merlin Tuttle is. Inform me. April 19th, everyone can get vaccinated. That's right. My three kids are now, they're all in their 20s. They're going to get vaccinated. They're happy. Yep. Let's see what else we have here. A lot of little one-liners. Today was the HIV lecture. So you can look at that one. It's the most up to date. Hasn't really changed much. Bear Grylls goes into caves. Where? That's a good question. That's interesting. Arturo mentioned that viruses tend to send the immune system into overdrive. I don't think that's a fair statement. I think some of them do, but not all of them do. Many of them are well behaved in terms of tickling the immune response. So I think the worst ones are the zoonotic pathogens that are brand new. Do I get it right? They detect emerging viruses from a surge in pneumonia. Well, they had a pneumonia screening program in place in China because after SARS-1, right, they said we have to look for severe pneumonia. And that's how they picked up these 40 first cases in Wuhan. Would it be widely spread already? I'm not sure widely would be correct. As I said earlier, it starts in a farmer and his family in the town and then goes to Wuhan. It could get bigger and bigger, but you might not notice it until then. Do our cells have the innate enzymatic machinery to integrate random foreign DNA inside the nuclei? Yes, they do. In fact, cells pick up DNA all the time. They don't like to have it in the nucleus. They don't like foreign DNA. There's systems to get rid of it. But sometimes DNA gets in and it integrates randomly little bits of DNA, and there are traces of viral DNA in all our genomes. You can look at the human genome sequence and find bits of all different viruses integrated harmless in harmless places. Could the vaccine DNAs integrate? Sure, in a muscle cell, and then the muscle cell's gone eventually. It doesn't matter. So I don't see the problem with that. What are the specific T cell epitopes? So a T cell epitope is some amino acids in length, and they're produced by chopping up the spike. in the cell, and then those epitopes are displayed on the cell surface, and a T cell can detect it and kill the infected cell. So they're part of spike. They're like 20 or more T cell epitopes on spike, and there are many epitopes on other viral proteins as well. There are 12 mutations in the variant. Coronavirus is killing young and children. Why? So you have no idea if mutations have anything to do with it, okay? You're putting two things next to each other that are associations, and children and young people die everywhere of coronavirus, so I'm not sure what special is about that. Tom says, oh, thank you for the super chat. And David Tuller's posts. David Tuller's been writing on my blog for years about ME/CFS, and now he's writing about long COVID. He's really good. He's a really good writer. So I suggest you go to virology.ws and check it out. I also write there about once a week. Both Fauci and Walensky say it's more transmissible. I disagree. Sorry. Are you going to do this kind of live stream for the Reddit, or are you going to type everything? I don't know. I thought an AMA was typed. Whatever they want. Where's that other question? Here we go. Fauci and Walensky say it's more transmissible. I think they're wrong. I think they're looking at the original data, which are flawed. Here's why. Just one part of it. They calculated the R0, the reproductive index, without taking into account population effects. And another study I saw where they said the 117 variant, the outbreak is no different than if there had not been a B117 variant, according to their calculations. So you see, these things are all done by statistical models. And so I want biological data, and there aren't any. So that's why I think they're wrong. All right, so yes, this guano scenario, wouldn't there be a trail of doctor visits? Yeah, but they don't know what's wrong with these people. They have no way of diagnosing. And if they're mild illnesses, now the WHO report didn't find any such trail in Wuhan, but they said they're going to keep looking. So maybe they'll come up with something. But you might not see it. I agree that it could leave a trail, but you just might not see it. Would it be more effective to use different types of vaccines, different proteins encoded by the mRNAs? Yeah, I've always thought that spike only was a bad idea, right? So it turned out to work, but now we're selecting for variants, and maybe that's because of the focus on spike. I always thought we should have more viral proteins. So if we make a second generation vaccine, I'd love to include mRNA for N protein, for example. Yeah, for sure. Let's see, what else do we have here? Which is bigger, this chat or a typical Cornell virology lecture? So I don't teach at Cornell, I teach at Columbia. But my class is 130 students. One year it was 170. So we have 553 people here. So this is bigger. I've always said that I can teach more people using social media than I ever have in all my classes, for sure. Yeah. And you people are all over the world. It's not just at Columbia, New York. So I will do this long after I stop teaching. Although I'd always like to give lectures. I think it's fun to make a lecture and give it and get questions, right? Yes, I think these people have different definitions of transmissible. I think that's part of the problem. I think a spread of a virus to the population can be by fitness, improved fitness, which doesn't mean droplets moving through the air. And I don't know what they mean. It's a good point. You've mentioned that coleslaw can contain plant viruses that are harmless to humans. What sort of viruses are frequently found in cabbage? Well, the one study I looked at is an insect virus of caterpillars, the cabbage looper insect virus. They crawl over the cabbage, and the virus contaminates the cabbage, and you can't get rid of it. So you eat it, and it just passes through you. And if you're interested, the most common human fecal RNA is pepper mild mottle virus. It's a virus that causes disease of pepper plants. And we eat a lot of peppers, apparently, and it goes right through us, this virus. Most common virus RNA in your stool. Let's see here. So we have 5 minutes. Let me acknowledge some super chats. We're always way behind in the chat. I don't know what to do. If you have any suggestions, thank you, Credulopathic Remedy, for your super chat. Well, that's the last. That's towards the end. What was I saying? There's a lag, and I don't know what to do with it, but I've reached the end here. We don't know any. There's nothing new on the clots in the AstraZeneca vaccine. Still looking at it. They're still looking at it. Okay. Well, it looks like you don't want to know how many viruses there are in your food. Hey, I've reached the end here. Look at that. I've caught up, which is unusual. Do you think that 1 year from now we will be back to normal? Yes, 1 year. I think the fall is going to be more normal. I agreed to a speaking engagement in April next year in Zurich. And so I think next April for sure. But I think the fall is going to be normal. I think we're going to have classes. John, I just have to object. You can't say it undoubtedly caused an increase. That variant appeared at the same time the increase was observed, and you cannot say that. I'm sorry. You may feel that way, and that's fine, but it's not about what you feel. President of Brazil does not care about COVID, gives the medical system no support. That's why it's so bad there. I've heard to that effect, yes. I'm sorry to hear about that. Coastal is good. Don't worry about the viruses in it. It's not a problem. I've heard that SARS-CoV-2 has used up most of its tricks when it comes to spike, now risking losing binding affinity. So there certainly is a mutational space that a virus occupies, and it can't exceed it. We don't know what that space is yet for SARS-CoV-2. I would not underestimate any virus and its mutational space. So, yeah, you may think so that it, and there obviously are constraints, right? You can't change so much that you risk binding to ACE2. But where that is, we don't know. We're not there yet. And that's why tracking the variants is important. Tracking them and understanding them is important. But putting headlines in newspapers about how they're going to destroy humanity is not important. It's a waste of time. I think it would be terrific if you would lecture the public on live stream. So I'm thinking maybe I teach a virology course on live stream, right? And you guys anybody can listen in and maybe post some questions. Just another way of teaching. So this thing is answering your questions, but a proper lecture where I show slides, and at periodic intervals we can have a quiz. You can ask questions. I'm thinking of doing that. You think that's a good idea? If you do, let me know. How long after SARS-2? That's a good question. It may have been, well, the virus may have arisen years ago and just needed to have an encounter with humans, or it could have arisen, weeks before or months. We just don't know. If a vaccine is given and it hits a blood vessel, is it still as effective in the muscle? No, you want the vaccine, the mRNA vaccines, to get in the muscle cells. And if it gets in the blood vessel, it's going to be cleared very quickly. It's not going to be as effective. So that's not a good thing. All right, folks. Super chat, Kathleen. Thank you very much. I appreciate it. And Frank P. We have to thank Frank P. for modding tonight. I really appreciate it because the tone of the live stream has been much better since Frank kicked out some people who were being nasty. There's no reason to be nasty if you disagree with me, right? And yes, okay, I'll do a live stream. And yes, they don't have agency. You're right. I even make a mistake on that from time to time because I'm trying to speak quickly, and sometimes you do that. You're absolutely right. No worries. No worries. Start an email list for those of us who would watch. Yes, so that's something I'll do in my new studio. I'll work on a new course and do a live stream, like once a year or maybe more. Would I grade on a curve? If you want to be graded, maybe people don't want to be graded. Is this the John Gruber from Daring Fireball? Wow. If it is, welcome. And good to see you. All right, folks. Thank you all for coming again. I appreciate your participation, your questions and comments. And we will see you next week at the same time. Amy and I will come back. Thanks so much and good night.